Hello, I'm Lennart van Winden, postdoctoral fellow under Dr. Thaddeus Diamandis at the Princess Margaret Cancer Center. And previously I did a PhD at the Netherlands Cancer Institute in the Netherlands. And today I'm excited to give you a snapshot of my project funded by the Brain Tumor Foundation, titled Global Proteomics Reveals Bidirectional Integrin Signaling in Glioblastoma as a Driver of Invasion. So glioblastoma is the most prevalent and deadliest form of brain cancer, with patients having a median survival of just 15 to 18 months after diagnosis. And one of the major reasons for glioblastoma's ferocity is its swift infiltration into healthy brain tissue. Notably, a recent publication showed that in patient tissue slides, cell migration of glioblastoma cells is directed outwards of hypoxic pores, so areas with low oxygen, which indicates that glioblastoma infiltration could be induced by hypoxia. So while this is an important observation, no specific and potentially actionable signaling pathways were associated with this phenomenon. So we therefore hypothesized that we could leverage hypoxia to identify specific and actionable signaling pathways used by glioblastoma cells to infiltrate healthy brain tissue. And to do this, we leveraged a collection of 17 patient-derived glioblastoma stem cells, also known as GSCs, sourced from our collaborators at the Nylander lab in Sweden. Each cell was cultured for 3 days under different oxygen concentrations, including 20%, 1%, and 0.2% oxygen. And then next, we extracted proteins from the GSCs and analyzed them using a highly specialized analyzer capable of measuring thousands of proteins simultaneously, allowing us to map activated signaling pathways in GSCs under hypoxia. And when we don't look at our data, it showed that a number of selected invasion-linked signaling pathways positively correlated with increased hypoxia, whereas proliferative-linked signaling pathways were negatively correlated with increased hypoxia. While this indicates that our data shows a concordance with what we already know from literature, we hadn't found any new and potentially targetable signaling pathways. So to investigate specific invasive signaling pathways linked to hypoxia in our dataset, our next step involved conducting an unbiased analysis, in which we look at a database of thousands of signaling pathways and whether they are associated with increased hypoxia or normal oxygen conditions. And surprisingly, in the right compartment, in which signaling pathways are associated with hypoxia, we found that multiple integrin signaling modules were significantly enriched. And to confirm what we're seeing was actually real, we leveraged the publicly available data from the study involving tissue from glioblastoma patients. And notably, we found that in 2 slides with hypoxic cores, there was a substantial overlap with integrin signaling, supporting our evidence that invasive integrin signaling is induced by hypoxic conditions in glioblastoma. So next, we wanted to know whether integrin signaling was also used by GSCs to infiltrate healthy tissue independent of hypoxic conditions. And in order to investigate this, we used a glioblastoma organoid model, which is a hybrid organoid or lab-made mini-brains infused with GSCs. And here I show microscopic images of 3 glioblastoma organoid lines that were analyzed with the organoid in red and the GSCs in green. And as you can see, U32, 35, and U30, 54 showed extensive infiltration of GSCs, while infiltration of GSCs was limited in the U31, 17 line. For all 3 GSC lines, we measured proteins using the same proteomic method as the hypoxia experiment. And for 2 GSC lines, we obtained spatial transcriptomic data, a method to generate RNA transcript levels, the genetic material that directly encodes the proteins of thousands of genes that can be spatially allocated across the glioblastoma organoid. So to investigate whether the glioblastoma organoid system is a good molecular model to study glioblastoma invasion, we quantified invasive and proliferative signaling pathways in glioblastoma organoids relative to organoids without GCs. So here we observed an overall increase, as shown with the red color, of invasive gene sets for all glioblastoma organoids, except for integrin signaling U3117, which is in line with its limited infiltration. So, and in addition, we observed a high overlap between integrin signaling and GSC presence for both spatial transcriptomic slides. However, overlap between GSC presence and hypoxia was lower, indicating that integrin signaling also acts as a hypoxia-independent driver of GBM invasion. And in the previous data, we concentrated primarily on outside-in integrin signaling. And while preventing downstream integrin signaling seemed promising in clinical trials, the results haven't significantly improved survival for patients. And because of this, our research has shifted focus to inside-out integrin signaling. And this is about how proteins from within the cell activate or deactivate the integrin receptor, affecting their binding with the extracellular matrix, and is a key part of how cells migrate through the tissue. And I like the following analogy, where a cell represents a climber on the wall. So to move over the wall or across the wall, the climber needs to detach his arms and legs from the wall and attach it to other parts of the wall. And the cell basically moves similarly and can detach from the extracellular matrix, so the glue-like material between cells, and stretch itself out to attach again to the extracellular matrix through intercellular activation of the integrin receptor. By understanding how this intercellular activation and deactivation of the integrin receptor works, we hope to find an alternative treatment strategy that could be more effectively reduce the spread of glioblastoma. So pursuing alternatives, we found that MEF4K4 potentially acts as a key regulator in the activation of moisin, so one of the ERM proteins capable of deactivating the integrin receptor. So given this, we hypothesized that inhibiting MEF4K4 could potentially stick the GSCs down to the extracellular matrix, indefinitely stopping its ability to move around. And to test this hypothesis, we treated 4 of our glioblastoma organoid lines with a MEF4K4 inhibitor. And remarkably, we observed that the GSCs in our glioblastoma organoids did not spread through the healthy tissue compared to the control condition, as you can see here in the microscopic images. And notably, treatment with the inhibitor showed a decrease in infiltration, a significant decrease in infiltration of GCs through the brain tissue in 3 glioblastoma organoid models, except the less invasive U3117 line. So to confirm whether MEF4K4 directly interacts with moisin and the integrin receptor, we argued that interfering with the integrin receptor binding to the extracellular matrix could potentially reverse the effects of MEF4K4 inhibition. And in an experiment, we administered the inhibitor treatment to GSCs both with and without the antibody, specifically targeting the integrin receptor in the presence or absence of MEF4K4 inhibition. So in the control conditions, top left and bottom left, where MEF4K4 inhibition was not applied, adherence GSCs displayed typical growth. However, upon treatment with the MEF4K4 inhibitor, so top right, a notable presence of substantial gaps emerged on the culture plate. And this observation suggested that these cells were unable to migrate. And importantly, we had previously determined that the inhibitor treatment did not affect cell viability. And subsequently, when GCs received treatment with both the inhibitor and the specific antibody, so the bottom right, we witnessed a rescue of the previously observed effect, with the cells returning to a control-like pattern. So, importantly, when we use the percentage of background or non-red color as a quantitative measure, MEF4K4 inhibition alone is significantly increased, so there's more background compared to the negative control, but not significantly different when the antibody is added. So taken together, these findings indicate that MEF4K4 inhibition is mediated through moisin and the integrin receptor binding with the extracellular matrix. So our lab has shown the potential and effectiveness of a MEF4K4 inhibitor in laboratory models. And the logical next step is to validate these findings in a glioblastoma mouse model that closely mimics the human disease. However, our expertise doesn't extend to mouse model studies, and setting up the necessary facilities is beyond our current capabilities. So to address this, I'm actively working on a collaborative effort with the Van Tellingen lab in the Netherlands. They are experts in testing new drugs in glioblastoma mouse models and use advanced techniques such as patient-derived xenografts and brain slice cultures to study how glioblastoma spreads. Their methods for visualizing the cancer's infiltration in the brain are particularly effective. And what's more, they also work with the same GC lines that we use, which means that their research will directly complement and enhance our own. This is a lab I worked closely with during my graduate studies. And while my focus wasn't on glioblastoma at the time, I witnessed the quality of the work with pharmaceutical mouse models of glioblastoma firsthand. So with our project at a point where the collaboration could be beneficial, I remembered the Van Tellingen Lab's capabilities. And on a recent trip to the Netherlands, I reached out and visited the lab to discuss the idea of a collaborative effort. So they were very enthusiastic about my ideas and open to a collaboration. So as we initiate this collaboration, the Amandus Lab will continue to investigate how MEF4K4 affects GSCs and look for potential weaknesses we can target with new treatments. I will be leading this collaborative effort at the Van Tellingen Lab, ensuring that any new insights gained at the Amandus Lab can be tested in the Van Tellingen Lab's mouse models. And finally, our collective aim is to work towards a new treatment strategy that one day could eventually be part of clinical trials, hopefully through the collaborative efforts of the University Health Network and the Netherlands Cancer Institute. So finally, I would like to express my gratitude to my principal investigator, Dr. Thaddeus Diamandis, and all my lab colleagues for their support. And I would like to thank our collaborators at the Nederlander Lab for providing us with the GSC lines. But I'm greatly thankful for the funders, especially the Brain Tumor Foundation of Canada for awarding me this fellowship and the CIHR for funding the lab, making this research possible. And finally, I would like to thank you for listening. And if you have any questions, please reach out to Sue so she can forward them to me, and I will be happy to answer any questions you have. Thank you.